Hi, my name is Cienna Slattery. This summer, I have been working in the Crane Lab at the Midtown Center for Childhood Cancer Research at Seattle Children's Research Institute, and I'm going to be presenting on my work with identifying the function of DSIG4 as seen in the tumor microenvironment of glioblastoma. So glioblastoma is the most aggressive primary brain tumor. It has a 5-year survival rate of less than 10%. And the tumor is actually able to integrate with surrounding healthy tissue, making it impossible to completely remove with surgery. On top of that, immunotherapies have failed to provide a survival benefit due to the blood-brain barrier, which tightly regulates what is able to enter and exit the brain, the immunosuppressive tumor microenvironment of glioblastoma, where the tumor cells are actually able to kind of trick the surrounding immune and non-immune cells into becoming pro-tumor. And as an immune-privileged organ, the brain is able to tolerate the presence of antigens without initiating an inflammatory response. So tumor-associated macrophages are the most abundant cell in the tumor microenvironment of glioblastoma. These are macrophages that the tumor has tricked into becoming pro-tumor. So they secrete immunosuppressive chemicals and chemicals that aid in tumor development. These cells, they play a key role in initiating and maintaining the immunosuppressive tumor microenvironment of glioblastoma, and they have been associated with a poor patient prognosis. So by researching TAMs, the Crane Lab hopes to identify factors shifting the TAMs into a pro-tumor phenotype, as well as those allowing TAMs to survive in the tumor microenvironment, in hopes of supporting immunotherapies focused on the elimination of TAMs. So the Crane Lab performed a single-cell RNA sequencing of TAMs to figure out the immunosuppressive factors in them, and found that protein DSIG4 was significantly overexpressed, which is interesting because protein DSIG4 has been correlated with a poor patient prognosis in glioblastoma. In a follow-up experiment, the Crane Lab added glioblastoma supernatants to macrophages in vitro and saw that DSIG4 increased expression significantly. So then our next question was, does the overexpression of DSIG4 result in changes in global gene expression? And my project focused on answering this question. To answer this question, I performed an mRNA analysis, and for our analysis, we collected mRNA from control human macrophages and from DSIG4 overexpression human macrophages. We then used nanostring technology, which is a new technology that uses molecular barcodes that code for individual strands of mRNA. We then binded those molecular barcodes to the mRNA, put it through a machine that quantified the amount of each individual molecular barcode, and then it normalized the data to give us relative gene expression levels. And two interesting changes we saw was an upregulation of glutamate synthase, or GLUL. What's interesting about this is that this codes for an enzyme that is involved in the creation of glutamine, which is an amino acid that in excess can be converted into a fatty acid and used for fatty acid metabolism. So by upregulating this enzyme, the TAM is increasing the availability of fatty acids to be used for fatty acid metabolism, well, energy. And we also saw a downregulation of glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, and this is an enzyme that is involved in the breakdown of glucose. So there are two types of metabolism. There's glucose metabolism, which is the fastest, and it provides the fastest energy, and there is fatty acid metabolism, which is a little bit slower, but it gets the job done. So since tumor cells grow super fast and require a ton of energy, they love glucose. So what the tumor cells are doing, they're telling the TAMs to downregulate this enzyme that breaks down glucose so that they don't eat any of the glucose and they save all the glucose for the tumor cells, and instead they go to an alternative metabolism. And this downregulation of GAPDH aids in tumor survival. And together, these two changes signify a shift away from the glucose metabolism into a fatty acid metabolism. So overall, our results show that DSIG4 plays a crucial role in the ability for TAMs to survive in the tumor microenvironment of glioblastoma. And due to DSIG4's crucial role, it is a strong candidate for further study. Future studies may include those examining other factors involved in shifting TAMs from a glucose metabolism into a fatty acid metabolism, examining other genes that examining other molecular factors involved in the upregulation of BSIG4 in TAMs. In future immunotherapies may include those directly targeting BSIG4, as well as those blocking the upregulation of BSIG4. And I would like to thank the Crane Lab for allowing me to complete this research, and the University of Washington Neurological Surgery Summer Student Program for making this internship possible. And I would like to give a special thanks to Dr. Ella Mogan and Sandra Ella Mogan for creating this wonderful program, as well as Sylvia Zavachin and Julie Gold for being wonderful mentors and doing all the background work, and Dr. Nicole Lieberman and Nicole Priestborn for working with me directly on this project.